All right. So we've got that started, and I'm also going to, we're going to be recording this and we'll post it again. And people will be able to review this. So good evening, everybody. I'm Dina Battle with KCCure. We are a evidence-based, patient-centered research advocacy organization. And in 2019, we launched our Chromophobe research program. And I want to give a huge shout out to Catherine Utemeyer, who really spearheaded that effort. And I'm really excited tonight to be able to welcome Dr. Martin Voss from Memorial Sloan Kettering. And Dr. Voss was our inaugural grant winner last year, and he is going to tell us a little bit about the research that he's doing. So with that, I'm going to go ahead and turn it over to you, Dr. Voss, and hopefully you can share your screen.
Okay, let's see. So let me see if I can, just let me know if my slides are visible in the right mode. You guys can see the slides okay?
I can see them. I don't see any concerns in the chat, so I think we're good.
Okay, terrific. So let's get started. So thank you, Dina, and thanks everyone for dialing in tonight. My name's Martin Voss. I'm a geomedical oncologist here at Memorial Sloan Kettering Cancer Center in New York, where I spend my entire day around kidney cancer. So my clinical focus, my clinical practice, all the patient care I provide here, but also my research in terms of drug development and biomarker studies is entirely dedicated to kidney cancers, and us having a very large referral base to our center. We see a lot of unusual kidney cancers here, rare subtypes, and I have a special interest in chromophobe kidney cancer. I'm sharing my disclosures here. And for tonight, what I want to do is provide a bit of a broader overview over systemic therapies and sort of the armamentarium that we have specifically in the metastatic space. So as a geomedical oncologist, I primarily see patients with advanced resected, but first and foremost metastatic cancers, and that certainly applies in chromophobe kidney cancer also. So I want to go through the various therapy options that we have available to our patients these days, and to try to provide a little bit of perspective on how the many drug labels that are out there for advanced cancers of the kidney apply to chromophobe kidney cancer specifically. And Chromophobe RCC has been in the metastatic space a success story in many ways and for certain therapies, and less so in other ways. And that's an area of intense research by our group here and others. The slide that you currently see, many of you may have seen, iterations of this in the past, provides an overview of FDA labels for treatments that are out there for patients with advanced cancers of the kidney. This includes adjuvant therapies. We have 2 drugs that are approved for patients with resected non-metastatic disease, but primarily medications that have been developed through the years to treat disseminated cancers of the kidney. And it's relevant to know that all of the agents that you see here and all of the pivotal trials that led to the approval of these drugs were conducted chiefly in patients with conventional kidney cancer or clear cell renal cell cancer, which doesn't mean that they're not available to use in less common kidney cancers, non-clear cell variants like chromophobe kidney cancers. It just means that we may know less about them. And through the decades, of course, many of these drugs have been studied in less common variants. And the common approach, and this is a slide that I've taken from a review talk that one of my colleagues, Monty Powell, gave a few years ago, is what we call the bucket approach. So it's easy to do a large randomized study in a cancer that is common with a lot of patients that are seeking out treatments and are interested to enroll in trials, but for less common indications, or for what we call orphan cancers with just simply less numbers in patients out there, it's more difficult to conduct prospective clinical trials. For kidney cancer, traditionally what has been done is to think about renal cell cancer in sort of 2 broader ways. Clear cell renal cell cancer or conventional kidney cancer makes up about 3/4 of the patients we see, and then the remaining subtypes are often summarized as non-clear cell RCC. And a lot of the trials that have been done through the years have been done, essentially studying agents or regimens that were successfully developed in clear cell kidney cancer, now in smaller trials that sort of bucket in all of the various kidney cancer subtypes, including chromophobe kidney cancer. So we call that a mixed bag approach. And the downside to that is that you don't really learn all too much about any one of these rare kidney cancers specifically, but you learn sort of more. You get a sense of how things might work or a signal. So if we go back to the timeline that I provided before, then what I've done here is to sort of broadly categorize this into eras of drug development. This is how the field has changed through the years. If you see on the left-hand side, the salmon-colored segment, this is before we had molecularly targeted agents. This is right around the time that we started to gain a better understanding of biology, but cytokines were really the only agents that were effective to treat metastatic kidney cancer of any type. And turns out cytokines at the time weren't very successful in non-clear cell kidney cancers. And the yellow area, now starting with the approval of sorafenib and sunitinib, is what marks the advent of targeted therapies. And targeted therapies, again, were first studied in common kidney cancer, but quickly investigated for clear cell variant and for non-clear cell variants, and have proven to be quite successful and efficacious, particularly in chromophobe kidney cancers. And we'll talk about that a little bit. Starting in 2015 with the introduction of nivolumab, a lot of clinical trials were launched to initiate immunotherapy strategies in advanced cancers of the kidney. And through the subsequent years, most of the trials all of us were doing at the time integrated some element of immunotherapy, now knowing that that was available. And again, the story starts with clear cell renal cell cancer, where this was found to be successful, but shortly thereafter, the first trials started to test these drugs in rare kidney cancer variants, and chromophobe kidney cancer patients started to be treated on and off clinical trials with these medications. So throughout this era, immunotherapies of various kinds was tested until, with the advent of the first combination regimen, ipilimumab and nivolumab, this most recent era of treatment starts. So, this is the combination era. Most of the studies that have since been conducted and the labels that you see have come through since here are combinations of often immunotherapies together with targeted agents, and you see several of them plotted here. So what I'm going to do is try to sort of take us through these different eras and just highlight what we know about chromophobe kidney cancer and what we learned through the years. And then the second half of the talk, I want to speak a little bit about work that we've done here at Sloan Kettering, looking into the immune biology of chromophobe kidney cancer specifically, and how that might be leveraged therapeutically. And we'll talk a little bit about the grant that we were awarded through the KCCure here, and also what it is that we're doing. So we don't have to go through all of these in great detail, but these are the type mixed bag or bucket trials that I mentioned before, the ones that are most commonly cited because they are the largest trials done in non-clear cell kidney cancer in the targeted era before we had immunotherapies available, are ESPN, ASPEN, and RECORD-3. And these were studies that tried to compare the effectiveness of the main targeted strategies that were available at their time, to fight metastatic kidney cancer, and they compared VEGF targeted therapy, so blood vessel blockers, to everolimus, which is an oral medication that blocks mTOR. So it's an inhibitor of cancer metabolism. And on these trials, the same question was pursued in different ways. Is one of these strategies more effective than the other in the various clear cell, non-clear cell variants? And what I've done here is I've plotted the size of these trials so you get a sense, by comparison. Registration trials for common cancers often have many hundred patients, sometimes over 1,000 patients. And here, these are fairly smaller trials. The RECORD-3 was a bigger study, but included both clear cell and non-clear cell patients. And I've looked up the references, and I've put down exactly how many chromophobe kidney cancer patients were treated on these trials. And very quickly, it becomes apparent what the limitation is in interpreting this data. We can look at the outcome data. We can speak to the likelihood of response seen across these patients, but you can imagine, even without a PhD in statistics, that if you're only looking at 12 patients that were treated, 16 patients that were treated, 17 patients that were treated, there's only so much fidelity that you get out of the data in predicting for the next patient we see in clinic exactly how things are going to fare. Rest assured, what was interesting to see on the trial was that compared to all the other non-clear cell variants, chromophobe kidney cancer stood out in that both strategies turned out to be quite effective. So we have some non-clear cell variants where clearly one of the two is not effective. Typically, for most other variants, including clear cell, actually, angiogenesis inhibition, or these targeted agents that block blood vessel growth, tend to be more successful in terms of how long they control metastatic cancer, how likely they shrink metastatic kidney cancer, than these metabolite inhibitors of mTOR. And it turns out across all of these trials, there was a signal that said something is up with chromophobe kidney cancers in that they respond to both of these drugs, favorably, and maybe even better to the everolimus in some patients. And we know from studies of chromophobe kidney cancer biology that there's a fair proportion of patients for which the signaling pathway, the protein-protein interaction within and around the cancer cells, that lead to a misregulation of metabolism through this mTOR protein specifically, is oftentimes dysregulated through acquired mutations. So there are some indicators as to why chromophobe kidney cancers might actually respond better to everolimus than other variants. And the next step that sort of speaks to both of these being effective is that the combination of both strategies, was studied several years later. In this case, 2 oral medications, lenvatinib, again, an angiogenesis inhibitor, an oral medication that blocks vascular growth, and the same metabolism inhibitor, everolimus. So again, this was a bucket trial that was conducted much later and only presented in 2020, and it finally published in 2022. So it combined these 2 strategies in one regimen and looked at the various rare kidney cancer subtypes. You can see them color-coded here. So on the left-hand side is what we call a swimmer's plot. So what we look at are individual patients that go on to the trial and receive these therapies. And one of these lanes, or swimmer's lanes we call them, represents how one patient fared. And you can see at the bottom plotted out treatment duration month, how long the treatment was effective in these patients, meaning how long did they stay on therapy. If they have a little error here, then they're still on therapy at the time of analysis and so forth. And you can see that there are chromophobe kidney cancer patients here that have been on the same treatment for well over a year and remain on treatment and so forth. And again, the strongest signal was seen for this entity. Again, this wasn't a large trial. It accrued just under 10 chromophobe kidney cancers, but they tended to do well. And this is what we call a waterfall plot, which we do on clinical trials. We measure out the overall volume of metastatic disease and the CAT scans or MRIs that patients have every couple of months on these trials. And then we see, as they are on the study, does the cancer grow, in which case the bar goes up, or does the cancer, shrink over time, in which case it goes down. And again, you can see here highlighted in dark blue, the patients who tend to do very well here over to the right. There's quite a few of the chromophobe kidney cancer patients, that did very well. And the authors indeed highlighted, this is just pasted from their discussion, one of their main conclusions was particularly noteworthy in this study was the promising response rate of 44% and clinical benefit of 78%. I'm just going to give a little plug here and try to explain how we think through response data. I talk to my patients in clinic about this all the time. All of you do your best to keep up with the medical literature and read papers and so forth. A response rate of 44% does not speak to the fact that only 44% of patients on the trial had shrinkage in their tumors. And I'll give you an example. If you look at the waterfall plot, here are patients whose tumors clearly decreased in size and they may have done so for extended periods of time. In fact, they may still be on therapy with very good benefit. But in order to meet what we call an objective response, a certain % criteria has to be met. So there might be a greater proportion of patients who have tumor shrinkage. In fact, we can see here that 1, 2, 3, 4, 5, 6, 7 out of 9 patients with chromophobe kidney cancers, clearly their cancers are getting smaller on the treatment. It's just that a certain percentage of shrinkage has to be met in order to qualify for an objective response. So the clinical benefit rate here, meaning stopping tumors growth or shrinking things, was around 80%, and that stood out, in comparison to other therapies. And several of these patients had other treatments in the past. So this is a powerful regimen. It's considered by many, a gold standard now in patients with metastatic chromophobe kidney cancer. So clearly, targeted therapies, were proving to be successful. And I still remember the time when we were all attending conferences, back in the 2012, 2014 era, and we would talk in the non-clear cell sessions on the podium about the various kidney cancer subtypes and how they do. And people would ask, there was always the notion that chromophobe kidney cancers, whichever treatment we chose, mTOR VEGF inhibitor therapy, they did a little bit better than the other rare variants. So, they stood out to be benefiting from these then novel therapies more. Then we entered the immunotherapy era. I mentioned that before. So nivolumab, which many of you will have heard of, is a targeted immunotherapy drug that was FDA approved back in 2015, and it works by activating T cells and other immune cells in the tumor microenvironment. So there are cells that are sitting in and around the cancers and that have cytolytic capability, but they're dormant, they're sleeping, and they're inactivated molecularly through certain interactions that these tumor cells have with their surroundings, and the tumor cells in how they signal towards the microenvironment to tissues around them, can sort of switch the immune cells off. Now, nivolumab and other targeted immunotherapy agents that are out there reverse that process and can reawaken T cells in the area of the tumors and metastases. Now, after this approval came through, I told you many clinical trials started to explore other immune therapies and immunotherapy combinations across all types of kidney cancer. But the strategy for all of the drugs that have been in development, in fact, the ones that have since been approved, pembrolizumab, avelumab, ipilimumab, nivolumab, they all work by targeting and activating cytotoxic T cells or CD8 T cells. They do have effects on some other cells, antigen-presenting cells, and so forth, but ultimately, they're the executive cell that sort of brings about the cytolytic effect are what we call effector T cells. And this was tested after its success in clear cell kidney cancer in a number of non-clear cell kidney cancer studies. And much like before, these studies were only helpful to a limited extent because they tended to, allow for any non-clear cell variant to go in. So the largest one that has tested this strategy was Keynote 427, which was now published a year or two ago, and treated over 100 patients of non-clear cell variants with this drug called pembrolizumab, which is a PD-1 inhibitor. So it works just the way that I mentioned before. And this is taken from the original publication. Now you know what a waterfall plot is because we just talked about it here. You just have a lot more bars because it's more patients on this clinical trial. And again, we have this color coding to see how the various kidney cancer variant did. And if you remember, when we looked at the waterfall plot for targeted therapies, most of the chromophobe kidney cancer patients were towards the right-hand end where all the shrinkage is happening. And now if you look at it for pure immunotherapy with a PD-1 inhibitor, there are much lesser patients that clearly have benefit. There are patients, right? You see patients here that have deep responses to therapy, 1, 2, 3 patients. In this case, over 20 patients with chromophobe kidney cancer were enrolled on Keynote 427, and the majority of patients did not benefit from it. And if we look at this plot now on the right-hand side here, this is what we call a forest plot, okay? Some people call it a Christmas tree plot. And what we can do here is we can look at different subgroups within the clinical trial. These are all patients with metastatic cancer. They all have kidney cancer, and they all have clear and non-clear cell kidney cancers, but there are different variants that we can look at. And for the different variants, we can now look at the benefit. It looks at what we call objective response rate, and you see that little dotted line in the center. If something is right on the dotted line, then there's no then the objective response rate is met. That's a 30% reduction, so that's a PR. So you can see there are certain subgroups here that tend to go further towards the right, a higher response rate, 20, 40, 60, 80% response rate. And the chromophobe subgroup here is the one that is furthest towards the left. So of all the subgroups that we looked at on Keynote 427, PD-1 inhibition with pembrolizumab alone seemed to benefit the least proportion of patients in the chromophobe subgroup. Some benefit, but not many. And why is that? Oh, I should have pushed that button earlier. So but there you can see the objective response rate, the likelihood of actually having significant shrinkage is less than 10%, meaning deep responses, are not commonly seen. Now we have the combination era. So here we combine immunotherapies and targeted drugs, so combining the old and the new. And there was the question whether that was going to prove successful. So if you go through sort of the history of time for chromophobe kidney cancer specifically, success with the various targeted agents, mTOR inhibitors, and VEGF inhibitors, less so with immunotherapy. And now what about combination immunotherapy? This is Checkmate 920. So many of you will have heard of ipilimumab nivolumab as one of our most aggressive ways of trying to elicit an immune response. So here we give 2 immunotherapy drugs together. And in conventional kidney cancers and many other cancers, it had been demonstrated previously that that was more likely to induce a notable clinical response than just giving one of the drugs alone. So ipilimumab nivolumab is a standard of care in clear cell kidney cancer, and it was FDA approved in 2018. And there was the question as to whether if PD-1 inhibition with nivolumab or pembrolizumab alone was not so successful in patients with certain rare kidney cancers such as chromophobe, would it be helpful if we were more aggressive about it? Ipilimumab in its ways can actually redirect what we call naive T cells that don't know yet where, what type antigen they're going to fight, to find a new target, versus nivolumab, which really for the most part reawakens cells that are sitting in the tissue and technically already know what they're fighting. So there was the question whether that was going to be more successful. And the keynote, the Checkmate 920 study in cohort 2, which had different subgroups, looked specifically at non-clear cell kidney cancers, treated about 50 patients. And, I don't have graphics here, but none of the chromophobe patients that were treated on on Checkmate 920 achieved an objective response. So again, there might have been some patients who had some degree of shrinkage, but certainly not a clear signal that this was active or more active than PD-1 inhibition alone. And as we push the immune system more with combination immunotherapy like this, we do see more immune-mediated adverse events, and that was definitely seen on the trial. So we see more toxicity without an increment in efficacy. And here, this is actually a graphic from the presentation. So 7 patients with chromophobe kidney cancer treated in this 52 patient cohort, and the response rate for chromophobe kidney cancer was 0%. Our group has conducted a clinical trial that was recently published that now combines immunotherapy and targeted therapy, right? So the question is, what if we try to enhance an inflammatory signal in the tissue by using one of these VEGF inhibitor therapies? Might that make concurrent immune therapy more effective? And we had designed this trial to specifically look at different cohorts. There was a cohort of papillary and unclassified kidney cancers, and there was a second cohort that was meant to be 10 patients of chromophobe kidney cancer specifically. And these trials oftentimes have what we call a futility rule built in, meaning as we bring on more patients to the trial, we regularly check the data to make sure that the regimen is active enough to keep going with the study. If we learn from the trial 2/3 into it that there really is no effect, should we then bring on more patients? So this study actually closed its cohort for chromophobe early because we did not see effectiveness with this specific combination that was sufficient to keep going. This is the waterfall plot looking at cohort 1 and then cohort 2 over here. And arguably, there is some biologic effect, right? There is some shrinkage that is seen for these chromophobe kidney cancer patients that went on the trial. Remember, they were treated with 2 medications, so it's impossible for us to say, you know, what was going on here. But by comparison, certainly there's much a bigger effect for the other non-clear cell variants. And the impression, much like for the other immunotherapy-based strategies, was that this was not the way to move forward. So at the end of things, we have learned that immunotherapy, the way we apply it as a strategy in conventional kidney cancers, is just not the way to go for chromophobe RCC. Many have talked about this. I'm sure you've heard it, you know, through other channels in the past. And there's 2 ways to go about this. We can say, all right, we intensify our efforts in molecularly targeted agents, and that's just going to be the way forward, as it has been and remains effective. So for instance, cabozantinib plus nivolumab, in this regimen, we give the cabo at a lower dose. So maybe that's why we don't see a lot of shrinkage here. Maybe we should treat all of these patients at full dose cabozantinib from the get-go. Or secondly, we can say, all right, well, immunotherapy is relevant and has really changed the way we think about other cancers. Maybe it's just not the right immunotherapy. Maybe there are immune cells that we can activate in different ways. And that's something that we've been very interested in with my various collaborators here. And that I want to talk a little bit about. This is data that we've previously published a couple of years ago, trying to look into the molecular underpinnings of this disease. And again, it's a little bit of a blast from the past, or maybe a historical perspective, because this dates back to the targeted therapy era. So I told you, molecularly targeted agents, the drugs that are effective for chromophobe kidney cancer, they go after certain signaling pathways, certain ways by which the chromophobe kidney cancer cell itself affects its neighboring cells, be it other cancer cells, be it blood vessels nearby, be it fibroblasts or immune cells. And we had learned, and when we analyzed tissue from chromophobe tumors, that there was a high proportion of patients whose cancers had dysregulation in the PI3 kinase pathway, which is the pathway that is targeted by these metabolism inhibitors, offering somewhat of an explanation why these might work well. And we learned that there's a lot of chromosomal abnormalities in these tumors. The way we used to do investigations at the time was not very focused towards the immune system yet, in part because immunotherapies were just emerging as new strategies for cancer. And then secondly, also because the technology wasn't there yet to really dissect the immune phenotype in the lab. The computational methods weren't there yet. Things were incredibly expensive at the time. And since then, we've come a long way. And here's recent data, and this is how we now, think through characterizing the immune microenvironment in tumors. I have a close collaboration with the lab of Ming Li, who's a Howard Yu investigator here at the SKI at Sloan Kettering's immunology program. So we have laboratory scientists at our center here who are not seeing patients and may not even be working with human tissues. Maybe they're just modeling things in the tissue culture or in animal models and so forth, but all of their thinking revolves around immunotherapy. And we were very fortunate that this immunotherapy laboratory here gained an interest in chromophobe kidney cancer. And they did so because there's something unique that they found when they analyzed tissue. When we dissect human tumors and we look at them, what we can do with modern technology is we can, on a cell-to-cell basis, extract information from individual immune cells that are sitting inside and around the tumors, and we can characterize them. This is from a publication that was put out last year. And the data that you see in here is integrating 2 tumors, one clear cell tumor and one chromophobe tumor. And the different colors highlight different types of immune cells that we can describe by sort of trying to tease apart exactly what type what type of molecular programs these tumors on their inside have activated. And we can try to characterize the immune cells to see what type of immune cells are there and what is it they are up to. We can use computational methods to say, okay, there are certain immune cell types. So for instance, there are the type cells that modern immunotherapies tend to activate. And then there are rare immune cells that we think are left unaffected by the types therapies that are already FDA approved. And we were interested to look at this because we wanted to try to tap into untreated immune cells in the tumor microenvironment. It turns out chromophobe kidney cancers actually have a lot of immune cells in their tissue. So it's not like they're what we call entirely cold tumors or empty tumors that have no immune cells present. They have an abundance of immune cells in certain areas. But what was found in this analysis when looking at how that immune composition was made up was that this differed greatly between clear cell and chromophobe kidney cancer. In other words, it's just other types of immune cells that are sitting in the tissue here. So a different way to break it down. So the interesting clusters here are the biggest ones, the blue cluster, the orangish cluster, and that yellow cluster. And you can see here, these are the type cells that we target with PD-1 and CTLA-4 inhibitors. And they are predominantly present in the clear cell kidney cancer, but there are very few of these dormant T effector cells that nivolumab, ipilimumab, pembrolizumab would activate in chromophobe kidney cancers. Looking at this, it's actually not surprising that these drugs don't work because there's so few cells in the immune microenvironment of chromophobe kidney cancers that could be activated by the drugs that we commonly use for clear cell kidney cancer, where there's tons of these cells. So, not a big surprise that these can be leveraged. Now, it turns out that if you look at chromophobe kidney cancer, there actually is an immune compartment, which is this blue one that is very present in the chromophobe tumor and was not present to a great extent at all in the clear cell cancer. And we can now go about trying to characterize that a little bit more. Here you see the presence of the type cells that we find in clear cell kidney cancer, the type cells that respond to conventional immunotherapies. And if we look at peripheral blood, the healthy kidney of patients, and the kidney tumor of patients, then you can see in clear cell kidney cancer, there's few of these cells in the blood. There's more of these cells around the kidney, and there's a ton of these targetable immune cells sitting in kidney tumors. If we look at chromophobe kidney cancers, it's all sort of about the same. There's no real trend here. There's no statistical significance whether you have more or less of these immune cells that we target with pembrolizumab, nivolumab in chromophobe kidney cancers. If we look at this other subgroup, these new cells that were not previously defined, there's a very clear trend now for the chromophobe subgroup, whereas the kidney cancer by percentages, the conventional kidney cancers are over the map. And if we now try to look at this thing, we can characterize these new cells, and we can define them with surface proteins, and we can do these beautiful images. This is now, a magnification of a chromophobe kidney tumor. And we do superimposed immunofluorescence staining to highlight where these cells are, and you can see there. For the figure, they put little white arrows on you so we can see these cells. So it's these red little cells that are sitting all over the tumor. There's like these red immune cells that are sitting in there. And the question that came up in this context is, are these cells that we can target, just like the cells that we target with nivolumab that are not present in chromophobe kidney cancers? Okay, so what's their biologic function? It's nice that they're there, but if they don't have the capacity to kill cancer cells, then they can't be leveraged as a therapeutic modality. What's their clinical relevance in patients? So now that we know they're there and we can visualize them, measure them, maybe quantify them, how does that track with outcome? And then for me as sort of a clinician or a clinical trialist, it's always relevant to think about biology in a clinical context. And it's a very different clinical context, biologically speaking, and in terms of the immune biology also, looking at a tumor that is sitting in the kidney and has not had access to the circulation versus a tumor that is ever present in distant metastases. This is old data from 2019, and this is Sloan Kettering data here also. Josefina Casuselli is actually a urologist, and she was a visiting fellow at the time, and she looked at all the tumors within the surgeon's data bank here that had been resected over a number of years, and she compared outcomes for chromophobe and clear cell kidney cancer. So the blue patients, over 3,000 patients with clear cell or conventional kidney cancers who'd had nephrectomies at our center versus about 500 with chromophobe kidney cancer. And you could see if you just plot, their outcomes, that in the non-metastatic setting, meaning patients who go for the surgery, chromophobe kidney cancer patients tend to do better. Their likelihood of the cancer recurring is lower than it is for conventional kidney cancer. That's actually been known for quite some time. And it has to have something to do with the biology. So if you look at tumors of the same size with these two different entities, the likelihood of recurrence is lower for a patient with chromophobe RCC than for clear cell, and that was reflected in the Sloan Kettering data set. Now, if we now think about this and try to look back at these new immune cells that we found, then we can look at that risk of recurrence or the outcome for patients separately for clear cell and non-clear cell. There's something called the Cancer Genome Atlas Project, which was funded heavily by the US government through the National Cancer Institute several years ago. And it was essentially an effort, much like the Human Genome Project, if you remember at the time. It was an effort to try to get all the genomic and molecular details for different cancer cell types. And there were grants put out and a lot of funds given to study a big number of tumors from each relevant tumor variants. And the data molecularly was put into a repository that's publicly accessible. So it helps us when we study cancer biology to go to the TCGA data sets and apply molecular metrics. So in this particular case, our colleagues here in the immunology program had found these cells in chromophobe kidney cancers that we could now characterize them. They had done a lot of work trying to explain exactly how they look molecularly and how we can find them. And now we can go back to the TCGA data sets, which are hundreds of tumors that have previously been molecularly annotated and described. So that data is sitting on a server for everyone to look at. And they're annotated with clinical outcomes. So the TCGA for clear cell kidney cancer, for instance, is a couple of hundred kidney tumors, and the data de-identified for each patient includes, the sex of the patient, the age of the patient, when they did their surgery, how big was their tumor, but also did the cancer come back? Did they not come back? Is the patient still alive? Is the patient doing well? And so forth. So we can now use the molecular data and that clinical data to see what is the relevance of these cells. So if we use the expression signature that we found for these ILC1 immune cells, and we plot them for clear cell kidney cancer, then we actually find fewer patients that have a lot of these cells. But if they have a lot of them, they tend to do worse. They don't do better. For chromophobe kidney cancer, it's the exact opposite. If patients in their tumors have a signature that reflects a lot of these immune cells' presence, then their likelihood of the cancer coming back was much, much lower. And there's a lot of additional work that the group put into characterizing this, but ultimately it left us with the impression that these cells serve a real function in the tumor microenvironment, at least for primary tumors, before they metastasize. We can say that the tumors that have a lot of these ILC1 cells present, even if they had the same size as patients who had less of these cells in the tumor, were less likely to recur. And it suggests that they have a real immune surveillance function. The cells have a protective feature in that if there were a lot of these cells present, meaning the patient also had a lot of these cells elsewhere in their body, they actually did immune surveillance and prevented the chromophobe kidney cancer from doing harm, whereas they had no such role in clear cell kidney cancer, where patients didn't do better if they had a lot of these cells. The question is, can we somehow prove that these cells have a cytolytic function? And it had been proposed that cells of this type, because the immune cells, although we newly found them in chromophobe kidney cancer, had been described in other settings, other cancers, and so forth. And it had been suggested that the stimulant, the messenger substance that is used by the human immune system to activate specifically these immune cells, is a cytokine called interleukin-15 or IL-15. So all of us, as part of our immune response to fevers, flu shots, infection, or cancer, use a whole host of different immune cells in our defense. And those different immune cells signal through the circulation by using cytokines as messenger substances. They're kind of like hormones, if you will, that tell the immune system what to do. So if I, cut myself, dirt gets in the wound, and the immune cells in my hand are trying to recruit more cells from the lymph nodes and so forth to come and help fight the bacteria, they use cytokines such as interleukins in order to activate the immune system systemically and locally in the area of the infection. So IL-15, it turns out, seems to be the master regulator for these specific cells, these ILC1 cells that we think have relevance in chromophobe kidney cancer. And indeed, when our lab here studied this, they were able to show that the activity level and the ability of ILC1 cells to kill other cells, pathogens, or likely also tumor cells, was increased by giving the cells exposure to the cytokine IL-15. So it demonstrating that did two things for us. For one, it showed that these cells can be stimulated to be more active in a cytolytic fashion, meaning there are cells here, ILC1 cells, that have the capacity to kill other cells. So they're effector immune cells, indeed. And then secondly, there's a substance, IL-15, and that can be used in order to make them more active. And I'm not going to go through all the experiments that were done, but typically what immunologists do when they find something like this, they study it in different model systems in order to prove their point. So in this case, it was studied for different cancer models, kidney cancers, breast cancers, and so forth, in tissue cultures, and you see some images here, and also in mouse models. This is mouse data. What we can do with mice is, for one, we can replicate tumorous growth, and we can also take certain things out of the equation to see how relevant it is that they are there or not. So what you see here, for instance, these are different tumors in different mice, and there are mice here. These colorful bars represent mice that have been genetically engineered to not be able to produce this exact immune cell. So they can't have NK cells, they can't have ILC1 cells. And you can see that a mouse that is incapable of mounting this immune response with this particular immune cell, because they don't have these cells, they don't make them, are much more likely to grow tumors and metastases all over the body, in the liver or in the lung, than mice that are injected just with control and have an immune system that still contains these ILC1 cells. The second graph here shows that you can actually shrink tumors in mice. So this is kidney cancer tumors that were produced in mice by injection, and you can see if you give them control substance, sort of like giving placebo on a clinical trial, the tumor continues to grow, versus the next tumor in the same mouse that is treated with IL-15 or an IL-15 agonist is significantly smaller, has not grown. So what is it that we are doing as next steps here? And some of this work is being funded by your mechanism, by KCCure, and we're very grateful for that. We are studying these immune cells in patients, and we're studying them more for every patient that we see in clinic who has chromophobe kidney cancer, is being offered to participate in what we call tissue procurement protocols, where we say, okay, if you had surgery with us here in the past, are we allowed to access that tissue to study it? Or the next time we do a blood draw, can we do an extra tube and study certain things? If we ever send a patient for biopsy to confirm metastatic disease or metastatic tumor is resected, can we use some of your tissue, to study how kidney cancer, once it's spread from the kidney to other places, behaves differently? Because arguably the immune system is everywhere in our bodies, but it doesn't behave the same way in every organ, in every structure, and so forth. So there's a lot of studying the immune cells and how the progenitors of the immune cell looks like. The grant that Dina mentioned before that we were awarded last year looks at how these ILC1 cells expand in the peripheral blood of patients. It's not easy to get to tumor tissue. It's quite easy for us to do a blood draw. And immune cells, we argue, that are relevant for cancer surveillance are likely not only present in the tumor tissue, but they must come from somewhere. So we know that the progenitors, the cells that sort of give rise to these cells, cells that are immature and then turn into these ILC1 cells, they start in the bone marrow, they travel the body, and then they ultimately become tissue resident. And we can test peripheral blood of patients for the progenitors of these cells. And we have done, this is actually some of the samples that we tested in the context of that grant. So we can look at patients that have chromophobe kidney cancer. We can look at patients who have metastatic chromophobe kidney cancer, or we can look at healthy controls. And we can now try to see in the peripheral blood, do we see more of these ILC1 progenitors? We see a lot of these in patients who have chromophobe kidney cancer that is only present in the tumor. We tend to see less of them in patients with metastatic chromophobe RCC. So you can interpret that in many which ways. You can say, for instance, well, maybe there's less of these cells in the circulation, and maybe the cancer in some way suppresses the whole ILC1 mechanism. And because it's suppressed, the tumors now can metastasize and can grow elsewhere. And the next step could then be, well, if we could reinvigorate this progenitor pool to go back to the 88% that we saw in patients whose cancer is not metastatic, well, maybe we can go back to controlling the cancer, fighting the cancer, the way it must be controlled in early stage, because we know that chromophobe kidney cancers, once they're, actually have a low risk of metastasizing compared to other kidney cancer. Maybe that is because for most patients, this system remains quite active. And then once it somehow is dysfunctional, the cancer can escape this immune surveillance to these ILC1 cells and become metastatic. The other thing that we have started to do is to test IL-15 not only as a substance. You can buy IL-15 or you can generate IL-15 exogenally, but there are now companies out there that are actually developing IL-15 agonists or IL-15, what we call super agonists. So there are drugs out there that very potently stimulate the IL-15 receptor on immune cells. It's kind of like giving a synthetic version, of it in order to leverage the same molecular mechanism and the same aspect of the immune system. So we now have an agreement with 2 different companies to study their drugs in chromophobe model systems specifically. So what we often do when we have promising data like this, we take it to companies, we present the data, our data to them, and we say, do you have an interest to work on this angle with us? So we have 2 companies now that essentially let us use their agent, which is already in human development in other cancers, and let us test it in chromophobe models in mice in this case, and then in human samples. In this case, we can sort of slice pieces of human tumors. When a kidney tumor gets resected, we can put fresh tumor into a system that sort of keeps the cells alive, and then we can test directly what the effects are of these medications. And obviously the ultimate goal here is to do a clinical trial, and we are intensely working towards an IL-15 directed clinical trial specifically in chromophobe kidney cancer, and again, have definitely interest with partners here in the pharmaceutical industry to develop that. And if we achieve to do so, that would be the first chromophobe-directed clinical trial, the first trial that would study a strategy specifically in chromophobe kidney cancer, not as a bucket trial that puts in however many patients we can, get from one versus another type cancer. And also based on everything I just told you, with a rationale that was developed in this entity molecularly and by immunophenotype, not just replicating what's been done in clear cell kidney cancer. See, does this also work in chromophobe RCC? So with that, I'm gonna close. I'm almost at time here, but I'm happy to go overtime if you have questions. And we'll thank you all for for staying up late and listening to my monologue here.
Dr. Voss, that was amazing. And I mean, just thinking about the fact that we could have a clinical trial on Chromophobe, I mean, that makes me emotional. I think that's amazing. So thank you for your work. Thank you for everything you're doing. If you're able to stay on, we do have a few questions.
Yeah.
I did want to ask one question. You talked about how hard it is to get tumor tissue. Is that something that we can help you with? Could patients send tumor tissue to MSKCC? And you know, could you receive it? What form would you need to receive it in?
So you cannot send tissue to us to be studied without any type of patient relationship here. So we can't accept tissue donations for patients that have not been registered to our center because there's no mechanism for us to then have it processed. There are patients who never come to see a surgeon or a medical oncologist here who have their tumors reviewed as what we call a pathology second opinion. So a path second opinion is something that actually pathologists in smaller institutions commonly do. So if if you're a pathologist at a smaller size hospital that is not a freestanding cancer center, then the pathologists that examine any type operative specimen, they have to know all of pathology. They review benign findings, they review the appendectomy specimen, and they review the colon cancer, breast cancer, and their kidney cancer. So if something funky comes their way, they commonly send tissue samples to large academic centers like ours to ask, as the pathologist, for a second opinion. So I sometimes see patients in my own practice who come to see me with a clinical question, and I open their chart, and it turns out 3 years ago they already had a path second opinion when their kidney tumor was first resected, and the pathologist had sent the tissue here. Our pathologist weighed in, and they never saw a clinician. And now there's a new clinical question, say a metastatic treatment decision is to be made. Now they come to us. So, so to get back to your question, it is possible, to request a pathology second opinion and have your doctor send tissue alone to Sloan Kettering. And once that is the case, you're in the system, things are registered, and then there's a path forward. We do see patients, here for second opinions, even if they are, doing well with their treatment, happy with their care, and don't necessarily want to bring all of their medical care here. And certainly once we, interact with the patient in a, doctor-patient capacity, then it's very easy for our offices to sort of, talk to you about tissue consent and getting some slides from a prior biopsy you had and so forth.
Okay. So I'm gonna we have a few questions. I'm gonna allow people. So we're gonna start with Katie Coleman. Katie, can you? I think I just gave you.
There we go. I had a question on the slide that you presented that had the testing for the IL-15. It was image B, I believe, that showed shrinkage in the mouse model that I was curious what the subtype was for that mouse model, and if it was clear cell, if the results surprised you based on the TCGA data that showed clear cell had worse clinical outcomes with that. So the tumors in that report that we showed the response to were chromophobe kidney cancers specifically, and also there was some of the liver metastases data was actually from a breast model, because it turns out that these ILC1 cells are also relevant in other cancers. So it's interesting to me that, and I told you that immunology lab that works on this with us is not only focused on kidney cancer, but they, based on the signal that they saw for chromophobe, including the tumor shrinkage in the mice, they came to us. But for the breast model, I thought it was interesting because they were looking at this and one other cancer also that specifically metastasized to the liver, and they saw that these ILC1 cells are particularly prevalent in hepatic metastases or liver metastases in those tumor entities. They have not tested that yet for chromophobe kidney cancers, but I know
through my own practice that that's actually one of the more common places that chromophobe kidney cancer can go to. In fact, through the years, I've had several patients who, for some reason, only have hepatic metastases, not anywhere else. And it's interesting to me to hear them say, "Oh, there's something unique about these cancer controlling immune cells, specifically in hepatic metastases." So one other thing that we actually have started to do, which I didn't show here on the slide, and we're trying to get grant funding for that presently, is a lot of the work that we've seen, most of what I've shown for kidney cancer, that shows this very strong signal for chromophobe kidney cancer is early stage chromophobe. And I think it is relevant for us to know exactly what happens with metastatic chromophobe kidney cancer, ILC1 cells, IL-15 regulatory signals, and the potential to target them, especially because I'm interested to, set up this clinical trial. We obviously want models that demonstrate this to be efficacious, not only in models of primary tumors, but also metastases.
Thank you. That's
Okay.
actually very interesting.
So our next question is from Chase Griffith.
Hi, thanks, Dina. It looks like you're in the dark. I don't know what.
Yeah, it's just a lighting issue.
Okay, just wanted to make sure the timeline is different. Dr. Voss, thank you so much for taking time here. One of the questions I had was if the ILC1 showed up in other cancers. I think you answered that it shows, somewhat in breast cancer, some others. So I guess I'll skip to my next question, which
I can just, sorry to interrupt you, Chase, but the one point I would make, and all of this you always have to take with a grain of salt, but based on the computational comparisons we can make from like the very from the various TCGA models that are out there for like all the big cancer types, chromophobe kidney cancer really stands out as to how many of these ILC1 cells we see, just more so than in any other cancer types that we've looked at here, which is not to say that it can't be relevant elsewhere, but it truly stands out. These are very infiltrated tumors, and for some reason, and you saw that comparison clear set to chromophobe, it's like the vast majority of the cells that we saw in the samples that we've looked at are these ILC1s, and we're trying to look at more.
My question would be too, what would be the potential timeline on any clinical trial you might have? A lot of people here, myself included, don't have a lot of experience on the trial, how that works. So if any chance of giving us that. And then do you know of any other IL-15 clinical trials that are going on elsewhere, even if it's not chromophobe specific?
Yeah, that's a good question. So there are a number of IL-15 agonists and super agonists that are in clinical development in humans already. So they are in clinical drug development. There's no kidney cancer specific trial and there's no chromophobe specific trial, but there are all solid tumor trials. But typically, industry sponsors start their endeavor with a new molecule by doing what we call all solid tumor trials or multi-solid tumor trials, where they allow for this. There's IL-15 drugs that have shown promise in lung cancer trials, and oftentimes you will see lung cancer trials, breast cancer trials, prostate cancer trials early because there's so many patients. So it's easier for any company to set up the trials, and there's more for them to gain. Companies are always interested in the big indications because if they show that something is effective, it's always the hope that it's the home run. So so there is, and there was recently a publication in New England Journal Evidence, which is a subjournal of the NEJM, that looked at a clinical trial in bladder cancer specifically, where an IL-15 agent was given for patients with early stage bladder cancer as something that they instilled into the bladder, and they could demonstrate there that the likelihood of the cancer causing recurrent problems in the bladder significantly went down, and there was a lot of interest in the GU oncology community in that drug, and it's one of the drugs that we test here in our preclinical system. So that drug, is in the two drugs that you see here on the slide are in clinical development across a number of different cancers, and have in the past been tested in all solid tumor model systems. When these drugs come out, there's often early stage investigations where the companies look at like various ways to give them intravenously, subcutaneously, into the bladder, and so forth. So there's a lot of soul searching that companies do early on, but it's fairly easy for you to find. There's review articles out there if you go, even on Google Scholar and so forth, and you can look, IL-15 to treat cancer, or IL-15 agonists in human cancer, clinical development of IL-15 superagonists, and so forth. You will see that there's 3 or 4 drugs out there that are in phase 1 testing at this point. And if you go to clinicaltrials.gov, then with the names of these agents, you can seek out where these trials are accruing patients. And typically, clinicaltrials.gov for the different sites actually has contact information on there. So that's a way to find these. If not, I'm sure, I know, the KCCure team is very well versed in these questions also. They, they can give you some pointers, I'm sure.
Martin, are you okay to keep going for a little bit longer?
Yeah. Mm-hmm.
Okay. So next, I want to give everybody a chance if we can. We have Marilyn Harris.
Hi, can you hear me?
Yep.
We can hear you, yes.
Okay, thank you so much for this program. I had some specific questions about, does the size of the tumor matter to towards metastasizing chances? How about necrosis inside? Looking specifically as well, do you test the immune system healthiness before you do trials so that you know how damaging it is to the immune system in general? Those are my 3 basic questions.
Okay, good. Yeah. So the first question, Marilyn, is, about risk stratification in patients who have their tumor resected, right? So you're saying, is there something that we can find out about the size of the tumor necrosis and so forth that tells us about the likelihood of tumor recurrence or the risk of the cancer coming back in the future? And the size of the tumor is one thing that has proven somewhat prognostic across all kidney cancer variants. So we know that the bigger the tumors get, the more likely they gain access to lymphovasculature and other circulatory elements, in and around the tumor, in and around the kidney, and that translates directly into the risk of recurrence. I mentioned this before, the same size, of a chromophobe versus a clear cell kidney cancer doesn't confer the same risk. There are different nuances that go into the models that we have out there, but that's a relevant one. Tumor necrosis, I think, is less relevant in chromophobe kidney cancer. We don't tend to do a lot of it, and the few models that we have to prognosticate risk of recurrence don't really take those into account as sort of a strong prognostic factor. And then you asked about fitness of the immune system, or other factors that we might use to predict tolerance of immunotherapy. So generally speaking, we use certain clinical criteria to speak to the risk of having adverse immune effects. We know that patients whose immune system baseline outside of their cancer is overactive to the extent that it's caused other issues, in your health before, should probably not go on immune activating therapy, specifically if they've had bad problems with their immune system. So if you, prior to your cancer diagnosis, have had a problem with an immune system that is overactive the way it is in patients with lupus or systemic psoriasis or rheumatoid arthritis, that is really bad. And now I give you an immune activating agent, then there's the potential that, while I might make the cancer better, I might make your other medical problems worse. So those are the best indicators for us that help us predict toxicity. There's no biomarker, there's no blood tests or tissue tests that we presently have in order to predict that. I'm actually doing a project with BMS, which is the company that makes nivolumab and ipilimumab, where we are using some of the metrics from patients, looking at their genomics and their tumor biology, to see who had toxicity and who didn't, and are there any molecular markers of toxicity, which is something that oncologists don't tend to investigate much in academic studies. So we're doing this big project trying to see if there's any type markers that we could identify before we put a patient on drug, in order to see if they're more likely to have toxicity or not. But that's ongoing work. We don't have a good marker yet.
Next, we have Mark Breyer.
Yeah, if you can hear me.
Yes.
Yeah, a general question with, given what you presented today, and thank you very much. What would you use tomorrow if someone had, and I don't have this, Dina, so it's a fair question. But
if I saw a patient in clinic tomorrow with newly diagnosed metastatic chromophobe kidney cancer, is that your question?
Yes.
So right now, I use combination targeted therapy as sort of the way to go based on what I've shown you before. Immunotherapy alone, which is what we use for many patients with clear cell kidney cancer, or even the combination of targeted and immunotherapy, I think does not add much in terms of efficacy, but does add in terms of toxicity. So I'm much more inclined towards combination targeted agents. So the lenvatinib everolimus combination is one that is very popular. There's a combination that I didn't show here that we have tested on a clinical trial here at Sloan Kettering, where that same metabolism inhibitor everolimus is paired with a drug called bevacizumab. And again, when we did that trial, there were some chromophobe kidney cancer patients who did extremely well. So I like the concept of combinations because you get at the cancer from two different angles. But right now, I don't use a lot of immunotherapy for the established immunotherapy drugs that are out there because the likelihood of getting a significant anti-cancer effect is not very high. I use these drugs. It's not like I would never use them, but it's not the first thing I go to. Typically, the first thing I go to are combination targeted agents. And keep in mind, I've shown you that data. There are some patients with chromophobe kidney cancer that can benefit from immune therapy. So arguably, some of these tumors probably have cytolytic T effector cells of the traditional type sitting in there, but it's just not a very prevalent scenario. So to just give everyone immunotherapy the way we give it to everyone with clear cell, in my mind, doesn't make a lot of sense. There are some other strategies that others are looking into. I know that Lisa Hensky has met with you guys in the past. So she's a colleague and friend in Boston who has a strong interest in chromophobe kidney cancer also. And they have gone down the route of metabolism intensely. I mentioned this before, right? If we say that traditional approaches, the way we use them for other cancer types, don't work well here, either we intensify our efforts in molecularly targeted therapy, or we try to find immune therapies of a different kind. So with what we're doing here, I'm going more towards the immunotherapy route. And Lisa's research has suggested that there indeed are targeted pathways around tumor metabolism that are left unexploited and could also lend themselves to drug development. There's ferroptosis, which is a certain type of cell death that is tightly regulated in normal human cells. And it seems that chromophobe kidney cancer cells are very susceptible to that. So if we could induce, tumor cell death through means of that mechanism, I think that would be, that would be great also. And it would seem that recent science from her lab points in the direction that that's another angle that we could take.
So we're going to do just two more questions. Katrinka.
Lightning round.
Yeah, lightning round. Katrinka.
Sorry about that. Thank you, doctor. That was really interesting. Yeah, I was just curious, have you broken down the chromophobe into, the various types of diseases that there are, like VHL or bird hog Dubé or anything like that?
Mm-hmm.
Have any studies been done for the specifics? Thank you.
So yes, there's some data on that, right? So I think what Katrinka is asking is we can be more sophisticated than saying just chromophobe, because chromophobe can arise as a sporadic tumor. There can be high-grade chromophobes with sarcomatoid features. There can be bird hog Dubé nascent chromophobes that are happening next to clear cell in the same patient and so forth. And some of this we're trying to get to. It seems that the very high-grade chromophobes that have a sarcomatoid component, in the area of that sarcomatoid component, where we see the cellular programs change dramatically, and that's been known for some time, there actually seems to be an off-regulation of these ILC1 cells. They seem to be less present in these highly aggressive areas around the tumor. And again, it begs the question whether it might be that these areas become so aggressive because the immune surveillance program in those areas is dysfunctional for some reason. Or is it the other way around? Like a lot of these things are like chicken egg type questions, right? Is it that, it's too aggressive and it sort of overcomes immune surveillance, or is it so aggressive because there is no immune surveillance in those areas of the tumor, right? But we are looking at these things and we are finding differences across the nuanced, variations of chromophobe kidney cancer itself.
Thank
And,
you so much.
And we have Courtney. Courtney, are you there?
Courtney's on mute, I think.
Okay. I have asked him to unmute.
Hey, how's that? Can you hear me, Dina?
Yeah, now we can hear you.
Okay, sorry, I was just laying praise on you and Megan and the doctor for organising this. It's been fantastic, thank you. Normally Chromophobe gets at least a minute of these presentations, so to have over an hour is just amazing. A quick question on the trial that you're looking at running. Have you identified a targeted drug at this stage that you would look to use for that trial?
Yeah, I mean, we're interested in the two drugs that I mentioned before, right? And typically the way this works is we start to work with the companies and we need to, demonstrate a signal in models, right? And we do that for one. Companies want that because they have to, get financial support in order to run a trial, and then we want it because we also want to feel right about putting patients on, on a trial. So right now we have an agreement with these two companies to study these two drugs in model systems, human model systems, animal model systems, and also induced model systems. And depending on sort of where things go with that, we are hopefully going to take this further to human studies. So
I think you mentioned, sorry to interrupt, I think you mentioned they were on the screen before, but I don't think they came up on my screen at least. So if you're able to share those two, that would be great. Thank you.
Yeah, of course. Mm-hmm. And I think we can. I think Dana's probably going to share the slides later also, right? You mentioned, I think, that the slides are going to be posted.
We are recording this and the slides will be available. And we can also follow up
Right.
and share information within the community as well.
Yeah, and typically what we do also, right? So because a lot of your questions sort of around the trial, you point to that direction. It's like, when would this trial be available? What is the trial exactly, right? So with some steps away, from that, but once we get more concrete with these clinical trial concepts, and certainly once we work towards opening the trials, for rare indications like this, we certainly make sure that advocacy groups and patient interest groups are aware of this, and this is what we've done for other entities in the past. So we would make sure that all of you find out, of course, if there were clinical trial for Chromophobe here or elsewhere ongoing.
Absolutely. Well, Dr. Voss, I can't thank you enough for everything that you're doing and for giving us your extra time, especially on a busy night. I want to make a very brief, quick announcement. This program we launched just in 2019. In our inaugural year, we raised $50,000. Last year, we doubled that to 100,000. And this year, we're already at 25,000 for the year. And we think that we can reach another 100,000 this year. And this funding really has, I think, launched so much attention and focus on Chromophobe. And even these small dollars, I think, make a big difference in this rare subtype. For anyone who is interested in working with us on fundraising, please reach out to Megan Griffith. She's going to put her email in the chat, but we need all the help we can get. And we'd love to see these trials move forward and see more research in this space.
Yeah, and I have to say, from the investigator's perspective, right, or from the researcher's perspective, the KCCure grant that you guys put out, and we were fortunate to get, and I can't stress that enough, is the very first Chromophobe specific grant that was put out there. I thought that was amazing, and I just really want to applaud everyone who's been involved in that. And it's just like you said, Dina, there's just been much more interest to Chromophobe as an entity, because we've known this is not a new kidney cancer subtype that was just discovered, last year. This has been, a topic of conversation for decades, but it's not been intensely studied. It's not been, targeted specifically with an investigational strategy. Yours was the first grant out there, and since then, there's another grant that was recently announced for Chromophobe kidney cancer. There's sessions at investigator meetings that we now have around Chromophobe. So there really has been an awakening of sorts, and I think that's just so justified and just very satisfying to see. And I want to thank you all for being a part of that.
All right, I think I froze a little bit, but thank you so much, and I'm so grateful to you for this. Thanks to everyone for participating, and we'll talk again soon.
Perfect. Thanks again for the
All right.
invitation.
Take care.
And thanks for all the great
Bye.
questions, guys. Bye.